This diagram outlines the basic steps in isolating DNA from clinical specimens. The first step, if necessary, is to separate white blood cells from red blood cells. This is important because white blood cells contain nuclei, whereas red blood cells do not. Next, the white blood cells or other nucleated cells are lysed, meaning their membranes are broken open to release the cellular contents, including DNA. Once the cells are lysed, the next step is to denature or digest proteins. This process removes the structural and enzymatic proteins that would otherwise interfere with the DNA. Following protein digestion, the DNA must be separated from contaminants. These contaminants can include proteins, heme from blood, or other cellular debris. After this, the DNA may need to be precipitated. Precipitation pulls the DNA out of solution so it can be collected and purified. Finally, the DNA is resuspended in a final buffer, which stabilizes it for downstream applications such as PCR, sequencing, or storage. In summary, 1. Separate white blood cells from red blood cells if needed. 2. Lyse the nucleated cells. 3. Digest or denature proteins. 4. Separate DNA from contaminants. 5. Precipitate DNA when necessary. 6. Resuspend DNA in buffer for final use. This sequence ensures that DNA is clean, stable, and ready for molecular analysis. Let's cover DNA and RNA isolation, purification, and analysis. We begin with DNA isolation methods. There are 3 common approaches. Liquid phase organic extraction, which often uses phenol and chloroform. Liquid phase inorganic extraction. Solid phase extraction, which binds DNA to a solid matrix for separation. Next is the isolation and purification of nucleic acids. The source material can be any tissue that contains a nucleus. Blood can also be used, but it must be collected in an anticoagulant. EDTA in a purple top tube is preferred. Importantly, whole blood should never be frozen, as this can damage the cells and degrade DNA. For storage conditions, DNA should be kept in TE buffer at 4 degrees Celsius for short-term storage and at -70 °C for long-term storage. RNA is less stable and must be stored in RNase-free ultrapure water at -70 °C. Because RNA is unstable, extra precautions are required to maintain its integrity. Moving on to nucleic acid analysis, the amount, quality, and molecular size of DNA or RNA can be determined using UV spectrophotometry, agarose gel electrophoresis, fluorometry, and colorimetric blotting. Finally, we focus on UV spectrophotometry for nucleic acid quantitation. Both DNA and RNA absorb light at 260 nanometers, while proteins absorb at 280 nanometers. For DNA, the concentration is calculated as absorbance at 260 nanometers multiplied by the dilution factor, multiplied by 50 micrograms per milliliter. One absorbance unit at 260 nanometers corresponds to 50 micrograms per milliliter of DNA. For RNA, the concentration is calculated similarly, except the constant is 40 micrograms per milliliter. One absorbance unit at 260 nanometers corresponds to 40 micrograms per milliliter of RNA. Concentrations are reported as micrograms of nucleic acid per milliliter of the hydrating solution. Let's review quality assessment, nucleic acid yield calculation, and restriction enzymes. We begin with quality assessment using UV spectrophotometry. The ratio of absorbance at 260 nanometers to 280 nanometers is used to measure purity. A ratio between 1.8 and 2.0 indicates good quality DNA or RNA. A ratio below 1.8 suggests contamination, usually from proteins or other molecules. Next, we look at calculating nucleic acid yield. Yield is calculated as the concentration of DNA or RNA multiplied by the volume of the hydrating solution. For example, if the concentration of DNA is 250 micrograms per milliliter and the volume of the solution is 0.1 milliliter. Then the DNA yield equals 25 micrograms. Finally, let us consider restriction endonucleases. These are bacterial enzymes, often called molecular scalpels, that cut DNA at very specific sites. They recognize short DNA sequences, most commonly 4 or 6 bases long, though some recognize sequences of 5, 8, or more bases. Importantly, these recognition sequences are palindromes, meaning they read the same forward and backward. The enzyme cleaves at or near these sites, which allows precise cutting of DNA for molecular biology techniques, such as cloning or analysis. This figure illustrates how different restriction enzymes cut DNA, producing either cohesive ends or blunt ends. The first enzyme is BAMHI. It generates cohesive ends with a 5' overhang. This means that after the cut, single-stranded overhangs extend from the 5' end of the DNA. The recognition sequence for BAMHI is GGATCC, and the cut occurs between the first 2 guanine bases. The complementary strand is cut in a staggered manner, leaving overhanging bases that can easily pair with matching sequences. The second enzyme is KPNI. It also generates cohesive ends, but in this case, the overhang is on the 3' end. Its recognition sequence is GGTACC, and the staggered cut produces single-stranded extensions at the 3' ends of the DNA fragments. These cohesive or sticky ends facilitate recombination, since they can form hydrogen bonds with complementary sequences. The third enzyme is HAEIII. Unlike the others, it generates blunt ends. Its recognition sequence is GGCC, and the enzyme cuts straight through the middle of the sequence on both strands. This leaves no overhangs, just blunt ends. While blunt ends are less efficient for recombination, they can still be ligated together with the right conditions. In summary, BAMHI produces cohesive ends with 5' overhangs. KPNI produces cohesive ends with 3' overhangs. HAEII produces blunt ends with no overhangs. This comparison highlights how different restriction enzymes create distinct DNA fragments useful for cloning and genetic engineering.